Hey everyone, tonight we're stepping into one of the most fascinating anomalies in all of human biology, a blood type that affects roughly 15% of the global population, that has shaped royal dynasties and medical history and ancient mythology. That is the subject of more conspiracy theories than almost any other biological fact, and that is, when you strip away the myths and the speculation, genuinely strange enough to be interesting entirely on its own terms. We're talking about Rh-negative blood. Before we begin, take a moment to subscribe, but only if you genuinely enjoy what I do here, and drop a comment telling me where in the world you're listening from and what time it is for you. Now dim the lights, get comfortable, and let's ease into tonight's journey together. There is something genuinely puzzling about Rh-negative blood, and the puzzlement starts before you get anywhere near the conspiracy theories or the ancient astronaut hypotheses or the royal bloodline speculation. It starts with a simple biological question. Why does this variation exist at all, and why is it distributed so unevenly across the human population? That question, asked seriously and answered carefully, turns out to be more interesting than most of the more dramatic stories that have been built around it. Blood typing is one of the most fundamental tools in modern medicine, and the system we're most familiar with, the ABO system, which divides blood into types A, B, AB, and O, was discovered in 1901 by the Austrian physician Karl Landsteiner, work for which he eventually received the Nobel Prize. But the ABO system is only one of many blood group systems that scientists have identified in the decades since. The human red blood cell surface is decorated with a complex array of proteins and sugars that vary between individuals and populations, and the Rh system, named after the rhesus monkey in which a related antigen was first identified, is the second most important of these systems after ABO. The Rh system is defined by the presence or absence of a specific protein on the surface of red blood cells called the Rh factor, or more precisely the Rhd antigen. If you have this protein on your red blood cells, you are Rh positive. If you do not have it, you are Rh negative. The distinction matters enormously in medicine. Transfusing Rh positive blood into an Rh negative person can trigger a severe immune reaction, and the interaction between an Rh negative mother and an Rh positive fetus during pregnancy can, without medical intervention, be fatal to the child. The Rh factor is not a minor biochemical footnote. It is a medically significant genetic variant that has real consequences for human health and survival. Our three companions tonight bring three very different perspectives to this story. There is Dr. Lena, a hematologist, a blood specialist, at a teaching hospital in Northern Europe, whose daily clinical work involves the Rh system, and who has spent her career navigating the territory between the genuine medical significance of Rh blood types and the extraordinary volume of misinformation that surrounds them. There is Professor Idris, a population geneticist at a university in West Africa, whose research focuses on the geographic distribution of genetic variants across human populations, and who has thought deeply about what the unusual distribution of the Rh negative variant tells us about human prehistory. And there is Marcelina, a historian of science in Argentina whose research traces the cultural and mythological meanings that different societies have attached to blood, and who has followed the story of Rh-negative blood from its medical discovery through its elaborate elaboration in popular culture with a mixture of scholarly fascination and wry amusement. The story of the Rh factor's discovery is, like most stories in the history of science, considerably messier and more interesting than the clean narrative of a single genius having a single insight. The credit for the discovery is conventionally given to Karl Landsteiner, the same man who discovered the ABO system 40 years earlier, working in collaboration with Alexander Wiener in 1937, when they injected rhesus monkey blood into rabbits and guinea pigs, and then used the antibodies produced by the animals to test human blood samples. They found that these antibodies agglutinated, clumped, the red blood cells of most human beings, but not all of them. The majority reacted. A minority did not. The minority who did not were the Rh-negative individuals. The immediate practical significance of this discovery became tragically clear within years. Doctors had been puzzled for decades by cases of erythroblastosis fetalis, a condition in which newborn babies develop severe anemia, jaundice, and sometimes fatal complications despite appearing healthy at birth. The condition seemed to cluster in families and seemed to worsen with successive pregnancies, but the mechanism was not understood. The discovery of the Rh factor provided the explanation. In cases where an Rh-negative mother was carrying an Rh-positive fetus, a situation that arises when the father is Rh-positive and the fetus inherits the Rh-positive gene from him, small amounts of the fetus's blood could cross the placental barrier into the mother's circulation during pregnancy or at delivery. The mother's immune system, encountering the foreign Rh antigen for the first time, would produce antibodies against it. In a first pregnancy, this typically caused no problems. The antibody production was not fast enough to affect the first child. But in subsequent pregnancies with Rh-positive fetuses, the mother's immune system was primed, and the antibodies she produced could cross the placental barrier in the other direction, attacking the red blood cells of the developing fetus and causing the hemolytic disease that had been killing and disabling so many children. Dr. Lena has treated patients with hemolytic disease of the newborn, and she describes the condition with the clinical precision of a physician who has seen what happens when blood incompatibility goes unmanaged. Before the development of treatments, first exchange transfusions in the 1940s and 1950s, then the revolutionary Rh immunoglobulin injection in the 1960s that prevents the mother's immune system from developing antibodies in the first place. Hemolytic disease of the newborn killed tens of thousands of babies every year and left many more with severe neurological damage. The development of the Rh immunoglobulin treatment is one of the great successes of 20th century medicine, a genuine triumph of understanding over a natural process that was causing enormous human suffering. She finds it quietly maddening that the medical reality of the Rh system, which is fascinating enough on its own, is so thoroughly obscured by the mythology that has accumulated around it. The naming of the Rh factor after the rhesus monkey has contributed in no small way to the mythology. The implication, which some popular accounts have been happy to exploit, is that Rh-positive people are more closely related to rhesus monkeys than Rh-negative people, and therefore that Rh-negative blood represents some kind of original or purer human genetic state, uncorrupted by primate ancestry. This is not what the science says. All human beings are equally related to rhesus monkeys. We share a common ancestor with all primates, and the Rh blood type system has nothing to do with that relationship. The naming was simply a naming convention that reflected the experimental history of the discovery. The rhesus monkey was the experimental animal that happened to be used in the work that led to the identification of the antigen. The name carries no deeper significance. Marcelina finds the popular mythology around Rh-negative blood one of the most instructive examples she's encountered of how a scientific discovery gets culturally processed. The scientific fact, a specific protein either present or absent on red blood cells with specific medical consequences, is real and interesting. But the cultural processing of that fact has added layers of meaning that have nothing to do with the biology, royal lineages, ancient aliens, special psychic abilities, resistance to disease, enhanced intelligence. Each of these additions tells us something about the cultural anxieties and aspirations that the discovery was used to articulate. None of them tells us anything reliable about Rh-negative blood. The distribution of Rh-negative blood across the world's populations is genuinely unusual, and it is this distributional anomaly, more than any other aspect of the biology, that has fueled the speculation about the origins and significance of the Rh-negative variant. The global average frequency of Rh-negative blood is approximately 15%, but this average conceals an extraordinary range of variation between populations. The highest frequencies of Rh-negative blood are found among the Basque people of the Pyrenees region of Spain and France, where approximately 35% of the population is Rh-negative, more than twice the European average and more than double the global average. High frequencies are also found more broadly across Western Europe, particularly in Spain, Portugal, Ireland, and the United Kingdom, where Rh-negative frequencies range from around 15 to 25%. Moving eastward across Europe and into Asia, the frequency drops considerably. Among East Asian populations, Chinese, Japanese, Korean, Rh-negative blood is extremely rare, occurring in fewer than 1% of the population. Among sub-Saharan African populations, frequencies are generally low, though there is considerable variation between groups. Among indigenous peoples of the Americas, Rh-negative blood was apparently absent before European contact, though this is complicated by the admixture that has occurred since colonization. Professor Idris has spent years thinking about what this distribution pattern means, and he approaches it with the caution that is appropriate when you're trying to read the deep history of a population from a single genetic variant. The first thing he emphasizes is that the distribution of any genetic variant in modern populations is the product of a long and complex history: migration, genetic drift, natural selection, admixture between populations, and that attributing the current distribution to a single historical event or cause is almost always an oversimplification. The Rh-negative distribution probably reflects multiple historical processes operating over different timescales. The Basque anomaly is the most striking feature of the distribution, and it has been the most discussed. The Basque people are remarkable in several ways beyond their blood type frequency. Their language, Euskara, is a language isolate, unrelated to any other known language family, suggesting that the Basques are the descendants of a pre-Indo-European population that was largely replaced elsewhere in Europe by incoming agricultural communities from the Near East during the Neolithic period. The genetic evidence broadly supports this picture. Basque populations show higher levels of ancestry from ancient European hunter-gatherers and lower levels of Neolithic farmer ancestry than most other European populations. This means that the Basque Rh-negative frequency is probably a reflection of the frequency that Rh-negative had in the ancient European hunter-gatherer populations, preserved in the Basque population by their relative genetic isolation over thousands of years. The question of why ancient European hunter-gatherers had such high Rh-negative frequencies in the first place is one that genetics has not yet definitively answered. The Rh-negative variant, technically the deletion of a specific gene called RHCE, or more precisely the absence of the RHD protein that this gene encodes, must have arisen by mutation at some point in the human past, and it must have spread through the population either by neutral genetic drift or by positive natural selection. If it spread by drift, the random fluctuation of gene frequencies in small populations, then its high frequency in some populations is simply the result of chance combined with the particular demographic history of those populations. If it spread by selection, because carrying the Rh-negative variant somehow improves survival or reproduction, then there must have been some environmental or biological pressure that favoured it. The selection hypothesis is attractive because it would explain the high frequency of what is, from a reproductive standpoint, a costly variant, The hemolytic disease of the newborn that the Rh incompatibility causes is a real reproductive penalty for Rh-negative women, which natural selection would normally be expected to work against over time. If the Rh-negative variant is nevertheless at 15% frequency globally, and much higher in some populations, something must have been balancing that reproductive cost. Various hypotheses have been proposed: resistance to certain parasites, resistance to certain viral infections, interactions with other genetic variants that confer benefits in specific environments. None of these hypotheses has yet been proven, but the biological puzzle is genuine. The connection between blood and royalty is one of the oldest metaphors in human culture, and the specific story of royal blood in the context of the Rh factor and related genetic conditions adds a layer of genuine historical drama to what might otherwise be a purely technical discussion. The most famous example is not actually about the Rh factor at all, but about a different blood-related genetic condition, haemophilia, that spread through the royal families of 19th and early 20th century Europe from a single source, Queen Victoria of Britain. Haemophilia is a genetic disorder affecting the blood's ability to clot, caused by a mutation in one of the clotting factor genes on the X chromosome. Victoria was a carrier. She had one normal copy and one mutated copy of the relevant gene on her two X chromosomes, which meant she did not have the disease herself, but could pass the mutated copy to her children. Her sons, who inherited the mutation, had haemophilia. Her daughters, who inherited it, were carriers like their mother. Victoria had 9 children, and through her daughter, Vicky, and her daughters, Alice and Beatrice, the haemophilia mutation spread to the royal families of Germany, Russia, and Spain. The most famous haemophiliac royal was Alexei, the son of Tsar Nicholas II of Russia, and his wife Alexandra, who was Victoria's granddaughter. Alexei's condition, and the desperate search for treatment that it drove, played a role in the rise to influence of Rasputin, whose apparent ability to ease Alexei's symptoms through what we would now call relaxation techniques gave him access to the imperial family that contributed to the political instability of the final years of the Romanov dynasty. The haemophilia story is not an Rh factor story, but it has become entangled with the Rh negative mythology in popular culture because both involve blood, both involve royalty, and both suggest that the blood of European aristocracy was distinguished from the blood of ordinary people in medically significant ways. The historical reality is considerably less romantic. Haemophilia is a debilitating and potentially fatal condition that caused enormous suffering among the royal children who inherited it, and the Rh factor is a blood-type variant with no particular connection to royal lineages, except insofar as the European populations in which royal families predominantly married, and which happen to have higher Rh negative frequencies than most of the world, also happen to contain more Rh negative individuals. Marcelina traces the history of the concept of blue blood, the aristocratic claim to a special kind of blood that distinguished the nobility from the commoners, back to the medieval Spanish notion of sangre azul or blue blood, which referred to the visible blue veins under the pale skin of people of pure Castilian descent who had not intermixed with the darker-skinned Moorish or Jewish populations. The blood was not actually blue. Venous blood is dark red, not blue, and the blue appearance of veins through pale skin is a trick of light scattering. But the metaphor was powerful and enduring, and it contributed to a centuries-long cultural tradition of associating blood with lineage, purity, and social status that created fertile ground for the later elaboration of the Rh negative mythology. The connection between the Rh factor and royalty in popular accounts typically takes one of two forms. The first is the claim that European royal families had disproportionately high rates of Rh negative blood, which may or may not be true. The data is not systematically collected, but which would not be particularly surprising given that European royalty married predominantly within the same narrow pool of European noble families, who in turn were drawn from the same Western European populations that have the highest Rh negative frequencies. The second is the claim that Rh negative blood is somehow ancestrally connected to ancient royal or priestly lineages, the Merovingians, the Anunnaki, the Nephilim of biblical tradition, a claim for which there is no evidence but which has proven extraordinarily resilient in popular culture. Dr. Lena encounters the royal blood mythology primarily in the form of patients who come to her clinic having read about Rh negative blood online and wanting to discuss what their blood type means about their ancestry or their special status. She handles these conversations with care because the patients are often genuinely curious and sometimes emotionally invested in the idea that their blood type marks them as special. Her approach is to validate the genuine interest. The Rh factor is genuinely interesting. The history of its discovery is genuinely fascinating. The geographic distribution is genuinely puzzling, while firmly redirecting away from the claims that have no scientific support. She has had this conversation many times. She will have it many more. The theories about the ancient origins of Rh negative blood range from the scientifically plausible through the historically speculative to the entirely mythological, and they are worth examining in that order, because the transition from the plausible to the mythological is instructive about how scientific uncertainty gets filled with cultural narrative. The scientifically plausible story begins with what population genetics can actually tell us. The Rh negative variant, the absence of the Rhd protein, appears to be relatively recent in evolutionary terms. Studies of other primate species show that all non-human primates have the Rhd protein, which means that the mutation that led to its absence in some humans occurred after the divergence of the human lineage. Estimates of when this mutation arose vary, but most analyses place it somewhere in the range of 35,000 to 200,000 years ago, a wide range that reflects genuine uncertainty in the dating methods, but that firmly places the origin within the genus Homo, and most likely within Homo sapiens. The geographic distribution of the variant, concentrated in Western Europe and particularly in the Basque region, suggests that it either arose in the European hunter-gatherer population or reached high frequency there through selection or drift. The genetic evidence connecting the Basque high frequency to ancient European hunter-gatherers, rather than to the Neolithic farmers who replaced them across most of Europe, is one of the more solid findings in this area. The ancient European hunter-gatherers were the descendants of the first modern humans to colonize Europe, who arrived from the Near East or from Africa via the Near East, somewhere between 45,000 and 30,000 years ago. If the Rh negative mutation arose in this population, it would have been carried with them as they spread across Europe. Professor Idris finds the ancient hunter-gatherer connection the most scientifically interesting aspect of the Rh negative story. It places the origin of the variant in the context of the broad genetic history of European populations. The complex replacement of hunter-gatherers by Neolithic farmers from the Near East that occurred between approximately 9,000 and 5,000 years ago, and the subsequent incursion of steppe populations from the Pontic-Caspian region that brought Indo-European languages and additional genetic change between approximately 5,000 and 4,000 years ago. The Basque people are the most genetically distinct remnant of the pre-Neolithic European population, and their high Rh negative frequency is a window into the genetic profile of a world that was otherwise largely replaced. The historically speculative stories move from this solid genetic foundation into terrain where the evidence becomes thinner. Some researchers have proposed connections between Rh negative blood and specific historical populations. The ancient Egyptians, the Sumerians, the Phoenicians, the Celts, based on a combination of genetic data, archaeological evidence, and historical records. Some of these proposals are more credible than others. The connection between Rh-negative blood and Celtic populations, for example, is consistent with the genetic evidence that shows elevated Rh-negative frequencies in populations of Celtic ancestry across the British Isles and France, and with the genetic relationship between Celtic populations and the ancient European hunter-gatherer ancestry that shows up most strongly in the Basques. The entirely mythological stories begin where the historical evidence runs out and continue into territory that the evidence cannot reach. The claim that Rh-negative blood is connected to the Nephilim of the Hebrew Bible, the mysterious giant beings described in Genesis as the offspring of the sons of God and the daughters of men, represents the point at which biological fact and ancient mythology have been combined in a way that neither the biology nor the biblical scholarship supports. The Nephilim are a subject of considerable scholarly debate about what the ancient text actually means, and none of the serious scholarship on the subject has anything to do with blood types. Similarly, the claim that Rh-negative blood is evidence of ancient extraterrestrial genetic intervention in the human species, that it marks the descendants of alien-human hybrids, is a hypothesis for which there is no evidence and that contradicts the straightforward genetic explanation for the variant's existence. Marcelina has traced the development of the Rh-negative mythology through its various elaborations in the second half of the 20th century, and she identifies the publication of several popular books in the 1970s and 1980s as particularly influential in establishing the specific claims that now circulate so widely. The claims were not invented whole cloth. They were assembled from existing mythological and pseudoscientific traditions, combined with genuine genetic data about the Rh factor, and presented in a way that made the combination seem more coherent than it actually was. The result was a mythology that has the superficial appearance of scientific grounding while departing from the science at almost every point. The hemolytic disease of the newborn that arises from Rh incompatibility between mother and fetus is one of the most poignant stories in the entire history of medicine. A story of how a fundamental biological mechanism can, under specific circumstances, turn against the reproductive process it is supposed to protect. Understanding it fully requires understanding something about how the immune system works and why the Rh factor matters so much to maternal and fetal health. The immune system's fundamental job is to distinguish self from non-self, to recognize the body's own cells and proteins as belonging to the self and to attack anything that it identifies as foreign. This distinction is accomplished through a complex system of surface proteins on cells that act as identification markers, allowing immune cells to recognize friends and attack potential enemies. The Rh factor, the Rhd antigen, is one of these surface proteins, and when an Rh-negative person encounters Rh-positive blood, their immune system identifies the Rhd antigen as foreign and mounts an immune response against it. In the context of transfusion, this immune response is the reason why Rh-negative patients must receive Rh-negative blood. Giving Rh-positive blood to an Rh-negative patient can trigger a potentially fatal hemolytic transfusion reaction in which the patient's immune system destroys the transfused red blood cells. In the context of pregnancy, the situation is more complex and more tragic. An Rh-negative mother whose fetus is Rh-positive is, in effect, pregnant with a being whose red blood cells carry an antigen that her immune system does not recognize as self. During the first pregnancy, the placenta typically prevents significant mixing of maternal and fetal blood. Small amounts of fetal blood cells can enter the mother's circulation, particularly at delivery, but in most cases the amount is too small to trigger a strong immune response before the pregnancy is over. The first Rh-positive baby is usually born safely, but the mother's immune system has now been sensitized. It has encountered the Rhd antigen, and it has memory B cells capable of producing anti-Rhd antibodies rapidly if it encounters the antigen again. In a subsequent pregnancy with another Rh-positive fetus, the sensitized mother's immune system responds much more rapidly and much more vigorously. The anti-Rhd antibodies she produces are IgG antibodies, a class of antibody that is specifically capable of crossing the placental barrier. These antibodies cross the placenta and attack the fetal red blood cells, triggering hemolysis, the destruction of red blood cells that can cause severe anemia, jaundice, brain damage, heart failure, and death in the fetus or newborn. The condition worsens with each subsequent Rh-positive pregnancy as the mother's sensitization deepens. Dr. Lena describes the development of the Rh immunoglobulin treatment in the 1960s as one of the most satisfying stories in 20th century medicine. The treatment, a preparation of anti-Rhd antibodies given to Rh-negative mothers after delivery of an Rh-positive baby or at specific points during pregnancy, works by destroying any fetal red blood cells that have entered the mother's circulation before her immune system can form memory cells against the Rhd antigen. It essentially prevents sensitization from occurring in the first place. The result is that the mother's immune system never learns to recognize the Rhd antigen as foreign, and subsequent pregnancies proceed without hemolytic disease. The treatment is simple, safe, and highly effective. Before it was developed, hemolytic disease of the newborn affected approximately 1 in every 200 births in Western populations. Today, in countries where Rh immunoglobulin is routinely available, it is rare. The evolutionary puzzle that the Rh incompatibility raises is a genuine one. If Rh-negative blood causes such a significant reproductive penalty for Rh-negative women in pregnancies with Rh-positive partners, how did the Rh-negative variant reach 15% frequency globally and much higher in some populations? Natural selection should, over time, work against any variant that reduces reproductive success. The fact that Rh-negative is common suggests either that it was selected for by some countervailing advantage, something that Rh-negative individuals gain that offsets the reproductive cost of incompatibility, or that genetic drift in small populations allowed it to reach high frequency before natural selection could reduce it, or that the selection pressure against it has been weaker in practice than the theory would suggest. Professor Idris approaches this puzzle through the lens of population genetics, and he emphasizes the importance of population structure, the fact that ancient human populations were small, relatively isolated groups in which random genetic drift could have large effects on gene frequencies. In a small population of a few hundred individuals, a random event, a founding effect, a bottleneck caused by disease or famine or environmental catastrophe, could dramatically increase the frequency of any genetic variant, regardless of its selective value. The high Rh negative frequency in the Basque population may simply reflect the particular demographic history of that population, a small founding group with higher than average Rh negative frequency, maintained by relative isolation over thousands of years, in which drift has operated more powerfully than selection. The claims about the special abilities and characteristics of Rh-negative people circulate in popular culture with a persistence that is inversely proportional to their scientific support. They include claims about enhanced psychic abilities and intuitive powers, higher intelligence, increased resistance to disease, unusual eye colors, enhanced sensitivity to light, a different body temperature, a different number of vertebrae, and a special connection to extraterrestrial beings. None of these claims is supported by rigorous scientific evidence, and most of them are not supported by any evidence at all. The resistance to disease claim is the one that has the most plausible biological basis, and it is worth examining carefully because it illustrates both the kernel of truth in some of the mythology and the way that truth gets exaggerated into something qualitatively different. There is genuine evidence that Rh blood type, both positive and negative, can interact with susceptibility to certain diseases. Studies have found associations between Rh negative status and reduced susceptibility to certain parasitic infections, including malaria in some populations, and between Rh positive status and certain cardiovascular risk factors. There are also studies suggesting interactions between Rh blood type and susceptibility to certain infections, including some evidence that Rh negative individuals may have some protection against certain viral infections. Dr. Lena is careful about how she presents this evidence because the associations that have been found are modest. They show statistical tendencies in large populations, not dramatic differences in individual outcomes, and because many of the studies are preliminary, with small sample sizes and methodological limitations. The picture that emerges from the literature is one of small, real, biological differences between Rh positive and Rh negative individuals that are consistent with the known biology of the Rhd protein and its role in immune function. It is not a picture of Rh negative superhumans with dramatically enhanced immunity. It is a picture of the kind of subtle, population-level genetic variation in disease susceptibility that characterizes all blood type differences. The intelligence and psychic ability claims are in a different category entirely. They are not only unsupported by evidence, but are conceptually incoherent. The Rhd protein is expressed on red blood cells. It is not expressed in the brain. There is no known biological mechanism by which the presence or absence of a red blood cell surface protein would affect cognitive function, intelligence, or psychic sensitivity. The claims appear to be based not on any biological hypothesis, but on the cultural tradition of associating special blood with special people. The ancient aristocratic mythology of blue blood and noble lineage, translated into the language of modern genetics. Marcelina finds the psychic ability claim particularly revealing of the cultural logic underlying the Rh negative mythology. The claim positions Rh negative people as more sensitive, more perceptive, more attuned to dimensions of reality that ordinary people cannot access, a form of the older aristocratic claim to special perception and wisdom that was assumed to accompany special blood. The content has changed, psychic ability instead of divine right, but the underlying logic is the same. Special blood means special people. The democracy of genetics, the fact that all human genetic variants are simply variants, equally natural, none more special than others, is not a story that this mythology has any use for. The body temperature claim, that Rh negative people have a slightly lower body temperature than Ra positive people, is one of the few physical claims that could in principle be tested, and when it has been examined, the evidence does not support it. Normal human body temperature varies between individuals and across the day, and in different conditions by ranges that are vastly larger than any reported Rh-associated difference. The vertebrae claim that some Rh negative people have an extra vertebra is equally unsupported. Extra vertebrae do occur in the human population, but their occurrence is not associated with Rh blood type in any study that has been done. The idea that blood type shapes personality is not primarily a Western phenomenon. It is most strongly developed in Japan and South Korea, where the blood type personality theory, known in Japanese as Ketsueki Gata, has been a significant cultural force since the 1920s and reached a peak of popular influence in the 1970s and 1980s that has not entirely subsided. The theory assigns specific personality traits to each of the 4 ABO blood types. Type A is organized and anxious, Type B is creative and selfish, Type O is confident and insensitive, Type AB is rational and indecisive, and it has been used in Japan for everything from personal compatibility assessments to hiring decisions to the organisation of workplace teams. The origins of blood-type personality theory lie in the early 20th century, when the scientific discovery of blood types prompted a wave of speculation about what other human characteristics might be associated with blood type. In Japan, a series of articles and books published in the 1920s by Takeji Furukawa proposed that the 4 blood types corresponded to 4 distinct personality types, drawing on both the Western scientific literature on blood types and the Japanese cultural tradition of categorizing people by temperament. The theory was initially dismissed by the scientific establishment but found a popular audience, and it was revived and elaborated in the 1970s by a journalist named Masahiko Nomi, whose popular book sold millions of copies and established blood-type personality theory as a mainstream cultural phenomenon in Japan. The scientific evidence for blood-type personality theory is, to put it charitably, weak. Large-scale studies of the relationship between ABO blood type and personality traits measured by standardized psychological instruments have generally found no significant associations. The apparent associations that people observe in daily life are almost certainly the result of confirmation bias, the tendency to notice and remember information that confirms our existing beliefs and to overlook information that contradicts them. If you believe that Type A people are anxious and organized, you will tend to notice when a Type A person behaves in an anxious or organized way, and to explain it by their blood type, while overlooking the many occasions when they behave in ways that do not fit the stereotype. There is also a social dynamic that complicates the assessment. In Japan, blood type is widely known. Many people know their blood type and know the personality theory associated with it. Research in psychology has consistently shown that people tend to behave in ways consistent with what they believe their type to be, a self-fulfilling prophecy effect that would tend to create real correlations between blood type and behavior, even if the underlying biological relationship is zero. People who believe that Type A means anxious may become more attuned to their own anxiety, may describe themselves as more anxious in surveys, and may behave in more anxious ways in social situations where their blood type is known. Professor Idris finds the blood type personality phenomenon interesting from a population genetics perspective, because the ABO blood type frequencies vary significantly between populations, which means that any personality effect, even a genuine one, would translate into different population-level personality distributions in different countries. If Type B is the creative and selfish type, then populations with high Type B frequencies, which tend to be found in parts of South Asia and East Asia, would on average be more creative and more selfish than populations with low Type B frequencies. This prediction is not just unsupported by evidence. It contradicts the kind of stereotyping and generalization about national characters that we recognize as culturally problematic when applied to other human characteristics. The Rh factor has been absorbed into the blood type personality tradition in Japan and South Korea to a limited extent, with some popular accounts assigning additional personality characteristics to Rh negative individuals beyond those associated with their ABO type. These claims are even less supported than the ABO personality associations, since the Rh factor adds another layer of specificity to claims that are already scientifically unfounded. But they illustrate the general principle that cultural traditions of meaning-making around blood have a momentum that carries them beyond whatever scientific content originally motivated them. Setting aside the mythology and returning firmly to the medical science, there are genuine and important ways in which Rh-negative blood type has significance for modern medicine, beyond the pregnancy complications we've already discussed. Some of these have direct clinical implications. Others are more subtle interactions with disease susceptibility that researchers are still working to understand. The most straightforward medical advantage of Rh-negative blood is in the context of blood transfusion. In emergency situations where a patient's blood type is unknown and there is no time to cross-match blood, the safest option is to transfuse O-negative blood that is type O in the ABO system, and Rh negative. O-negative blood is called the universal donor blood type because it can be transfused into any patient regardless of their ABO type or their Rh status, without triggering an immediate hemolytic reaction. This makes O-negative blood extraordinarily valuable in emergency medicine. It is the blood that is stocked on military helicopters and in emergency departments for use when there is no time for blood typing. The scarcity of O-negative blood is a genuine challenge for blood banking. Since only about 7% of the population is O-negative, the overlap of type O in the ABO system and Rh negative in the Rh system, the supply is limited relative to demand. Blood donation campaigns specifically targeting O-negative donors are a regular feature of blood banking in countries that maintain civilian blood supplies, and the military importance of O-negative blood has made it a focus of logistical planning in military medicine. The universality of O-negative as a donor type is, in a sense, a genuine medical advantage of being Rh negative, but it is an advantage for the recipients of O-negative blood, not necessarily for the O-negative donors themselves. The research on Rh negative blood and disease susceptibility is more complex and more tentative. Studies have found a range of associations between Rh blood type and various health outcomes, but interpreting these associations is difficult because blood type is correlated with many other genetic variants. It is not a single isolated gene, but a marker for a particular region of the genome that contains many genes, and the associations with disease may reflect the effects of neighboring genes, rather than the RHD protein itself. One area of active research is the relationship between Rh blood type and susceptibility to parasitic infections. There is evidence from African populations that Rh negative individuals may have some protection against certain parasitic infections, including some forms of malaria. The mechanism is not fully understood, but it may involve interactions between the RHD protein on red blood cells and the surface proteins used by malaria parasites to invade red blood cells. If Rh negative red blood cells are harder for malaria parasites to invade, this would provide a selective advantage for Rh negative individuals in malaria endemic regions, but it would not explain the high Rh negative frequency in European populations, which were not historically subject to the same malaria pressure as tropical African populations. Dr. Lena is cautious about the disease association research, not because she dismisses it, but because the field is at an early stage and the associations that have been found are often not replicated across different study populations. What she can say with confidence is that the RHD protein is a real biological molecule with real functions in cellular physiology. It is not just a surface antigen, but appears to play roles in cellular transport and possibly in other cellular processes, and that understanding its biology more fully is a legitimate and potentially medically important research program. The mythology has, if anything, hindered this research by making it harder to have a calm scientific conversation about what is actually known and what remains uncertain. After everything that the science has established about the Rh factor, its discovery, its medical significance, its geographic distribution, its evolutionary history, there remain genuine mysteries that the evidence has not yet resolved. These genuine mysteries are worth dwelling on because they illustrate that the real science of Rh negative blood is more interesting than the mythology, not less. The biggest genuine mystery is the selective advantage question. Why is the Rh negative variant at 15% global frequency and much higher in some populations when the hemolytic disease of the newborn that it causes in mixed-type pregnancies should, all else being equal, select against it over evolutionary time. Several hypotheses exist, as we have discussed, but none has been conclusively proven. This is a real open question in human evolutionary genetics, and the answer, when it comes, will tell us something important about the selective pressures that shaped human genetic diversity in the Pleistocene. A related mystery is the function of the RHD protein itself. We know what it is, a protein on the surface of red blood cells. We know its medical significance in the context of transfusion and pregnancy. But the full range of its biological functions in normal physiology is not yet understood. Recent research suggests that the RHD protein may play roles in cellular transport, in cell membrane stability, and possibly in interactions with pathogens that go beyond the simple antigen-antibody relationship that its medical significance emphasizes. Understanding the full biology of the RHD protein might shed light on why its loss in Rh-negative individuals has the population-level effects that the geographic distribution suggests. Professor Idris is most intrigued by what the Rh-negative distribution tells us about the population history of ancient Europe. The connection between Rh-negative frequency and ancient European hunter-gatherer ancestry is one of the clearest examples of a genetic variant that allows us to trace the demographic history of a population across thousands of years of migration and admixture. As ancient DNA sequencing becomes more routine, as it is now possible to sequence the genomes of ancient individuals from archaeological sites across Europe and Asia, the picture of Rh-negative frequency in ancient populations is becoming clearer. What is emerging is a more nuanced picture of how the variant spread and was maintained in different populations over the past 30,000 years, and this picture has implications not just for the history of the Rh factor, but for our understanding of European population history more broadly. The question of whether there are other populations with distinctive Rh-negative frequencies, populations that have not been as extensively studied as Europeans and Basques, is one that population geneticists are beginning to address as genetic data from diverse global populations becomes available. Early results suggest that the picture is more complex than the simple European-centric narrative of Rh-negative as a European or Western Eurasian phenomenon. There are populations in other parts of the world with elevated Rh-negative frequencies that are not well explained by European admixture, and these populations may hold important clues about the deeper history of the variant. Marcelina observes that the endurance of the Rh-negative mythology in popular culture, its resilience against the debunking efforts of scientists and science communicators, says something important about the human need to find significance in biological difference. We want our biological characteristics to mean something beyond their literal physical reality. We want our blood type to tell us something about who we are, where we come from, why we are the way we are. This need is not pathological. It is human. The problem arises when it leads us to accept claims that are not supported by evidence, and when those claims have real consequences, medical decisions made on the basis of misinformation, discriminatory practices justified by pseudoscientific race science, or simply a distorted understanding of our own biology. Dr. Lena, Professor Idris and Marcelina share a commitment to the idea that the genuine science of blood, the real history, the real biology, the real medical significance, the real evolutionary puzzles, is more interesting than the mythology. Not because the mythology is not fascinating in its own way, as a cultural phenomenon, as a window into human psychology and the history of ideas, but because the real story, when you follow it all the way from the discovery of the Rh factor in 1937, to the hemolytic disease ward in a modern teaching hospital, to the ancient DNA laboratory, reconstructing the genetic profile of European hunter-gatherers, is a story of genuine discovery and genuine mystery that does not need embellishment. The blood that connects us all is not a metaphor, or not only a metaphor. It is a physical reality, the red liquid that moves through every human body, carrying oxygen and nutrients and immune cells, maintaining the conditions that make life possible. And within that physical reality, the variations that distinguish one person's blood from another's, the ABO type, the Rh factor, the dozens of other blood group systems, are the accumulated record of a long evolutionary history, a history of mutations and migrations and adaptations and encounters that stretches back hundreds of thousands of years to ancestors who lived in worlds very different from our own. The Rh negative variant is part of that history. It arose by mutation in a population of ancient humans, probably ancient European hunter-gatherers, based on the best current evidence. It spread through some populations and remained rare in others, shaped by the forces of genetic drift and natural selection and population movement that govern the frequency of all genetic variants over evolutionary time. It interacts with the immune system in ways that have real consequences for transfusion and pregnancy. It may interact with susceptibility to certain diseases in ways that researchers are still working to understand, and it is present today at frequencies that range from virtually zero in some populations to over 30% in others as a living genetic record of a history that can now be read, with increasing precision from the DNA itself. Dr. Lena thinks about her patients, the Rh negative women who come to her clinic during pregnancy. Anxious about the incompatibility and what it means for their child, the Rh negative donors who give blood and whose O negative blood goes into the emergency reserves that will one day save someone's life, the occasional patient who comes in having read about alien bloodlines and wanting to know if they are really descended from the Nephilim. To all of them, she brings the same combination of clinical precision and human warmth, the same commitment to explaining what the science actually says and acknowledging what it does not say. The blood is real, the medicine is real, the mystery is real, the mythology is a different kind of real, a cultural construction, a human story, not a biological fact. Professor Idris thinks about the geographic distribution, the map of Rh negative frequencies that he has spent years helping to refine and interpret. In that map, he sees not special people and ordinary people, not the descendants of aliens and the descendants of mere humans, but the record of a world in motion, ancient hunter-gatherers moving into Europe from the east and south, carrying their genetic variants with them, Neolithic farmers arriving later and displacing them, diluting their genetic signature in most of Europe, while the Basques, in their mountain redoubt, preserved more of the ancient profile. Subsequent migrations, admixtures, selections, all of them leaving their marks in the current distribution of a blood group variant that a scientist in 1937 found unexpected and interesting. The map is a history. The blood is a document. The reading of it continues. Marcelina thinks about the stories, the blue blood of medieval Castile, the haemophiliac princes of Victorian Europe, the Nephilim and the ancient bloodlines, and the psychic Rh negative sensitives of the internet age. All of them are attempts to make biological difference meaningful in cultural terms. All of them tell us something about the societies that produced them, about the hierarchies they valued, the anxieties they harbored, the desires they projected onto the neutral facts of human biology. The stories are worth understanding not because they are true, but because they are human, because they represent the ongoing, irrepressible human effort to find meaning in the facts of existence, to make the body tell a story about the soul. The blood that connects us all is the same blood, the same evolutionary heritage, the same basic biochemistry, the same fundamental humanness. The variations within it, including the Rh factor, are the surface of a much deeper commonality. 15% negative, 85% positive, distributed unevenly across a world of 8 billion people who share the same recent common ancestor, who carry in their genomes the accumulated wisdom and the accumulated accidents of a lineage that stretches back into a past that modern science is only now beginning to read with any clarity. We're all the descendants of survivors. We all carry the marks of the pressures that shaped us. The blood in the end is the same. This has been Midnight Chronicle. Sleep well wherever in the world you are tonight,